I'm Dr. David Baskin. I'm a professor of neurosurgery at Houston Methodist Hospital, director of the residency program, and I'm also director of the Kenneth R. Brain Tumor Center. I want to talk to you today about a completely new technology, and we're going to start by a word that we invented called oncomagnetics. So what is oncomagnetics? It's the use of spinning magnets to produce oscillating magnetic fields to kill cancer. Pretty crazy idea. I want to talk to you about how this started in the petri dish, literally with cell cultures, and how it ended up in patients. So Aristotle, many centuries ago, spoke about the four major forces of the universe. And at that time, it was earth, air, fire, and water. But of course, we know that's not the case. We now know that the four important forces of the universe are gravitation, electromagnetism, short and more powerful nuclear forces. It's interesting that magnetism is 10 to the 36 times more powerful than gravity. Think about that. Gravity holds the planets and the universe together, but electromagnetism is that much more powerful. We tried to develop a new technology to treat brain cancer and other cancers, and thinking about the forces of the universe, we decided that perhaps we could use magnetism. So as you know, the mitochondria is the powerhouse of the cell. Lots of things happen in the mitochondria for energy. And a very famous man named Otto Warburg in 1931 won a Nobel Prize because he discovered that cancer has different metabolism. He actually thought that was the cause of cancer, but it really wasn't. It's just that cancer changes the metabolism to get the maximum amount of energy out for the energy that it puts in. So as a result, cancer uses the mitochondria in something called the electron transport chain, where electrons travel from one mitochondrial complex to another, and as they do that, energy is produced. As a result of them jumping on the electron transport chain, a lot of them fall off. These are free electrons, so they produce reactive oxygen species, mainly superoxide and hydrogen peroxide. It's interesting that these cancer cells have adapted to this, which they actually like a certain amount of reactive oxygen species in there. But the idea was, could we get those ROS levels so high that we would kill the cells? And another interesting difference is the fact that cancer cells have reduced antioxidant defenses compared to normal cells. So we came up with this idea: could we deliver powerful magnetic fields into the electron transport chain and knock electrons out? And as we did so, there would be increased levels of ROS, and the levels would get so high that they would be toxic to the cancer cell. And the idea is, well, that's great, but what about normal cells? Well, normal cells don't have high levels of ROS to start with, and they have very powerful antioxidant defenses. So the idea was, maybe we have discovered the Achilles heel of cancer, which is, of course, the holy grail for cancer research. So here's the idea: deliver the powerful electric field, knock the electrons off, cause an increased level of reactive oxygen species, and then you activate a bunch of other things, including apoptosis programmed cell death. So you activate this enzyme called caspase-3. This then destroys the cancer cells, tears apart the DNA, and the cells die. So that was the idea. So we spent a year and a half taking glioblastoma cells from patients, growing them in dishes, and applying here just a regular rotating magnet, and here different frequencies that we thought would kill the cancer cells. And we almost gave up. We had a lot of theoretical calculations, but here now suddenly, look at this. The cancer cells are exploding. They're blowing up. And this was the aha moment after a year and a half of research, saying, yes, we could take spinning magnets and use them to kill cancer cells, at least in a dish. Well, if that's true, we should be able to measure the production of reactive oxygen species over time. So in this set of experiments, we used the spinning fields and we measured ROS. And you can see over time there's a linear increase in the levels of reactive oxygen species. So indeed, the theory proved to be the case. But then we said, okay, well, what about programmed cell deaths? What about triggering apoptosis? Well, when you trigger apoptosis, this special enzyme called caspase-3 gets elevated. And sure enough, nothing happens for about 250 minutes as the levels of ROS build up. But then when you suddenly reach a critical level, boom, you see this very large elevation of caspase-3, proving that what we thought we were doing was actually what we were doing. We were elevating the levels of reactive oxygen species so high that we triggered programmed cell death. So now we did the critical experiment. Everyone said, that's great, that's a great theory, but you're going to kill normal cells. So we took normal human neurons and normal human astrocytes, put them in a dish, and applied these same types of stimulation that was killing the cancer cells. And what did we found? No effect. In fact, maybe the neurons did a little bit better, maybe not, but the point is the theory that they have low levels of reactive oxygen species and they have powerful antioxidant defenses proved to be correct because they don't get killed. They don't even get damaged. They grow just like they normally do. So this was another very exciting thing because obviously you could drop an atom bomb on these, but then you would kill everything around it. This looked like this was a truly selective way to kill cancer cells. So the next thing we did is the classic experiment called the clonogenic assay, where we grew cells in culture and we watched how many new colonies formed. And again, the same sort of thing happened. This is the control. The tumors are growing very well. And when we put in the oscillating magnetic fields, almost complete wipeout of all the colonies. Again, proving that we're not only killing cancer cells, but we're preventing them from growing and just knocking them dead. Now you say, okay, great. You're producing high levels of reactive oxygen species. They're toxic. If you put an antioxidant in the field, you should abolish the effect. You should prevent the whole thing from happening. So there's a powerful compound called Trolox, and we added them to the field. And you can see exactly what happened here is without the antioxidant, we're killing the cells. We're knocking them down quite a bit. But then when you add the antioxidant in, you basically have the same effect as with control. So this was another powerful documentation of the fact that we were truly using reactive oxygen species and that we were quenching the effect with an antioxidant just like normal cells do. Normal cells have levels of ROS, but they wipe it out with antioxidants. This slide is a very important slide because one of the challenges with glioblastoma is they're cold. And what do we mean by cold? We mean that somehow the tumors send out cells and molecules that prevent the immune system from coming in. We all have cells that mutate, but our immune system finds them and kills them. And one of the theories of cancer is that somehow these cancer cells prevent the immune system from doing their job. This has absolutely been the case with glioblastoma. Many of the checkpoint inhibitors that work extremely well with melanoma don't do anything because the glioblastoma cells produce an environment that prevents the immune system from coming in. So what we did here is we studied a variety of different cells and genes, and it turns out that as we kill these cancer cells, we open it up to the immune system. This is perhaps why this works so well, is now not only are we killing cells, but we're knocking out the defenses. We're knocking out the things that the cancer cell sends out to prevent the immune system to come in. So all of this stuff over here are a variety of cells and a variety of defenses that are trying to come in and kill these cancer cells, and now the gates open and they can come in. So this is a very exciting and important slide because it suggests that maybe not only are we killing the cells, but we're helping the immune system do the job that it so desperately wants to do. So now we switch to animal studies. You say, okay, this is great in a dish, but what about an animal? So we took a mouse that develops a glioblastoma, it's a GL261 model. We implanted them in the mice, and we started to treat them. And initially we saw, uh-oh, look at this. The tumors are getting bigger. But we remembered that sometimes in clinical studies, when you treat patients, there's swelling as your treatment is effective. So we persisted on, and sure enough, in a large number of animals, the tumor went away. And if you look over here, the survival curve in these animals significantly changed just with oncomagnetic therapy. And we were able to do this study because we have a 7 Tesla animal MRI here at Houston Methodist Research Institute, but you can see the rather dramatic shrinkage of the tumor in animals. This is another study about the immune system. This is a study using RNA sequencing, looking at sophisticated ways to see about how the genes that prevent the immune system from coming in change. And you can see a rather dramatic change in these pathways that prevent the immune system coming in, a study at the molecular level rather than the cellular level, demonstrating that we're really opening up the gates to let the immune system come in and kill the cancer cells that are left after we kill what we can with the magnetic field. So this is part one of the studies that I'm doing, talking to you about the basic science and the bench work that is going to lead to the bedside trials in patients. Thanks for your attention.